My name is Wolbachia Friedman. I am associate researcher at Princeton University, and today I'm going to tell you about a few protocols that I developed to transinfect, initially to isolate Wolbachia from the hemolymph of infected flies, and later on how you can inject that hemolymph into uninfected flies. So basically, this is a very simple protocol to isolate hemolymph of insects, and we use this device from Millipore that contains a filter that goes on top of an epidural. So you basically need to put, we start from around 100 flies, you anesthetize the flies, you put them into the filter device, you can just use a funnel, and our 100 flies will fit very easily on this device. And wash it with 70% ethanol. You just take the filter away. You can throw away the ethanol, and then after 3 times, your last wash you do with milky water.